Yeah, hello everyone. As you can see on the screen now, you can see our remaining fall schedule. So we have 4 excellent talks over 3 different dates that I recommend for you to put on your agenda. But I'm very excited to have Roberta De Vito present today. Roberta is an assistant professor in the Department of Biostatistics, and she's also a part of the Data Science Initiative at Brown University. Her research interests include latent variable modeling, Bayesian and parametrics, variable selection via sparsity priors, and her work is frequently motivated by applications in epidemiology and genomics. Prior to joining Brown, she did her PhD at University of Padua and a postdoc at Princeton. So thank you, Roberta, for joining us. We're all very excited to see you give your talk. So thank you.
Thank you so much, Jan, for this very nice introduction and detailed introduction, I will say. And thank you so much for having invited me. It's an honor to be in this, presenting also in this group. So today I'm talking about a little adventure, I will call, that started like during my PhD and like it's continuously right now during my faculty at Brown, and it's regarding the multi-study and cross-study machine learning techniques. So let me give you, first of all, some motivation behind my approaches and models. So suppose that you have multiple studies, as you can see in this picture here, coming from different population. And in all of these different populations, what you have is, of course, different sample size, so different subjects in each population, but you can measure the same variables. And I have some example regarding that. Some example could come from microarray gene expression data, where you have different population, of course, but you can measure the same gene expression across these different populations. Another motivation comes from nutritional epidemiological data, where you have, again, different population coming from different countries. Like here, what we have is 4 from United States and 3 from Europe, where you can find the same nutrients measured across these different populations. And another example comes again from gene expression data, but right now we can see that the population are exactly different tissue in which the same genes are measured. And this is coming from the GTEx consortium. So in all of these examples, like there is one big question. How can we handle multiple populations and multiple studies? And statisticians are trying to solve this issue. And one immediate way to do that is to stack all the study in one big data set. And remember this, we can do this since we have exactly the same variables. And to perform some standard techniques such as factor analysis or principal component analysis. And in this way, of course, we can measure some common shared component across all of these studies, but it is missing one piece. And this piece is the study specific component, because of course, all of these studies, all of these population can have a different heterogeneity, a different variation, coming exactly from the population that we are measuring. So maybe many of you know this standard factor model, but I want to give you a brief overview of what is the standard factor model. So the factor model is a dimension reduction tool that allows to decompose the observed variable in 3 different P's. Like 2 P's are coming from a factor component. So you can describe the observed variables through few latent factors, where lambda1 describe the coefficient, so how much each latent variables will influence the observed variables. And finally, we have the error component that is normally distributed. So as a consequence, in the standard factor model, you can decompose the covariance matrix with two different P's. As you can see, the P's with lambda that is coming from the latent component, and the other component that is coming from the covariance matrix of the error term. So right now, we have one problem in the standard factor model. The problem arises since this decomposition is not uniquely identified, and actually we can put many different orthogonal metrics here that will bring us to the same decomposition for sigma 1. And so statisticians try to solve this problem by imposing some constraint on the factor loading matrix. For example, in this occasion, what we are doing, we are imposing a block lower triangular matrix. So we are not estimating these triangular here, white triangular here, but we are imposing many zeros. So right now we are performing a factor model in one study, study 1. So supposed to perform S different factor analysis in each of your population. So again, what you can get is this lambda. This lambda is really important because again, it measures the relationship between this observed variable and the latent factor. And let's take a look at these metrics really in detail. So here I have estimated my factor loading matrix. Here what I have is my observed variable for the row. On the column, what I have is each latent factor in study 1. And here what you can see is that there are some black and higher rectangular and lighter rectangular. So higher will be the rectangular and higher will be the relation between each factor and each gene. So in this case, for example, we can define cluster of genes highly expressed in factor 1 and in factor 2 and so on. And we can perform these factor analysis in each of our study. And we can assess again for each study, like cluster of gene highly expressed. Where we want to arrive is something like that. We want to arrive to define common factors that are these factors in green that, as you can see, does not change from study to study. And we would like also to define study-specific factor to assess the heterogeneity across, for each of these population or study. And the idea, as you can see here, is arising from genomic, where generally people comparing different studies estimate a shared component. But also they realize that there could be some variation across different studies. And this variation can come from biological variation or also platform in which gene expression are measured. Of course, this idea can be, and I will show you later, can be applied to many different data sets where the aim is to capture shared component and also study-specific component. So for this reason, I developed this multi-study factor model. And the multi-study factor model is a generalization of factor model. As you can see here, each observed variable in each study can be decomposed in 3 different quantities. One that is coming from the common part, I will call reproducible part. As you can see here, we have this phi, that is our loading matrix that does not vary with S, does not vary from study to study. We have a latent component for that. We have a study-specific component that is lambda S, and this lambda S is the study-specific factor loading matrices. And of course, these vary from study to study, and it is associated to a latent component as well. And finally, I have the error term. And as a consequence of this model and of all of these assumptions, each covariance matrix in each study can be decomposed in 3 different parts. One that is related to the reproducible part with phi. One that is related to the study-specific component via lambda s. And finally, we have again psi s, that is the covariance matrix of the error term. So as the factor analysis, also the multi-study factor analysis has some identifiability problem and issue that we will solve in 2 different ways. First of all, like we will apply this block lower triangular matrix for the rotation. Again, here what we have is this white triangular that like I do not estimate, but I impose at 0. And this is for the common and for all the study specific factor loading matrices that I have in my model. So it depends how many studies I have. And there is a second identifiability issue in the sense that some component of the common can leak into the study specific component. And so to avoid that, I impose that the rank of all of these matrices together is a full column rank. What does it mean? It means that each column vector is linearly independent from the others. So in this case, I'm avoiding to repeat some information in the factor loading matrices. So the computation is through the EM algorithm. So the EM algorithm is an algorithm to estimate latent factors. So is especially useful when you have when you cannot observe some variables. And the multi-study factor analysis, of course, we have 2 latent features. One that is coming from the common characteristic, this F, and another that is again latent, a latent feature that is coming from the study specific. And so the EM algorithm proceeds in this way. Suppose that you will observe these 2 components. So we can write the complete log likelihood in this way, and we can then iterate this EM algorithm. So in the first step, we have the expectation step, that is the expected value of the complete log likelihood. And the second step is the maximization step. Of course, like with the multi-study factor analysis, we have many parameters. So sometimes we need some variation of the EM, and we decided to go with this ECM algorithm. The ECM algorithm is exactly the same for the expected value, but it's a little bit different for the maximization step. So the maximization step is divided in many different small steps. So one that is updating the one parameter conditional to the other remain to the previous step. So here what we have is at t+1, we are updating the covariance matrix of the error term given the other 2 parameters at the previous step, and so on for the other. So this algorithm has nice property because of course, like if it converges to a maximum, and second, it will increase the likelihood at each iteration. And the ECM algorithm preserves the same property of the EM algorithm. So I want to show you 2 different applications for this multi-study in order to get a better view. I don't know, there are any questions so far? Feel free also to interrupt me if you have any questions. And
there are no questions in the chat, at least.
So far, okay. So I can go with the 2 different applications for the multi-study factor analysis. The first one is in gene expression application. So what we have here is 4 different cancer data sets. Of course, with different sample size. Here, what I put is also the different platform in which gene expression are measured. And here it's the late stage of the patient. I will consider for now the genes involved in the immune system pathway. And we actually detect the reproducible factor, the reproducible pattern via AIC criteria. And the model also select one common factor shared among all of these studies. And we perform also a cross validation by keeping 20% of the unit for prediction. And so we are using 80% of our data for training. And we compare the multi-study factor model with the factor analysis to see how much we are gaining or how much we are losing. And the factor analysis is actually performed in two different ways. One way is stack all the data in one, as I showed you at the beginning. And the second one is performing factor analysis separately to each study. And here, actually, what I get is that the mean square error is 5% smaller in multi-study than in factor analysis after merging all the study in one. And the mean square error actually is smaller than 11% in the multi-study than in factor analysis after performing separately this factor analysis in each study. So we are gaining a lot in terms of prediction compared to the standard factor analysis model. So let's take a look to the result. Here what we have is this heat map, and as you can see in this black rectangular, I have the common factor component that, as you can see, this common factor, that is one, does not vary from study to study. It's exactly the same. And then I have different study-specific factors for each of the study. What I perform is I gene set enrichment analysis to see if this common factor is significantly enriched in a known biological pathway. And this common factor is actually significantly enriched in the innate immune system. That is a very well-known pathway for cancer. And I also visualize the correlation between the study-specific factor loadings. And I saw that for these two studies, I have very high correlation between 3 study-specific for one study with the other 3 study-specific for the other study. And actually I discovered that these 2 studies are exactly in the same platforms. So what I did is I reperform the multi-study factor analysis, and I discovered that these 2 studies share the 3 common factors. And again, the first common factor is exactly the same as the factor that I discovered before, and is again significantly enriched in this innate immune system. The other 2 are really different, and they were not enriched in any pathway. So our understanding is that these 2 common factors are shared because they are sharing the same platform. So it's not coming from any biological meaning, but it's actually related to the platform in which gene expression are measured. So this is one application where I show that these multi-study factor analysis can work and has a meaning, like a biological meaning for both the common and study-specific factor loading. I want to present another application in nutritional epidemiological data that is exactly the example that I showed you before. So here what we have, I have 7 studies. And again, like 4 are coming from United States and 3 are coming from Europe. And I have 23 nutrients that provides a good picture of the overall diet. And what my collaborator wants is to understand if this population shared some dietary patterns and if they also have some extra study-specific pattern for each population. And again, I arrived to this heat map. And what I discovered is that they actually, all of these studies, they shared 3 different common dietary pattern. And of course, these names are established from my nutrition collaborators. The first is called animal product and cereal, and the name is established from high level of total protein, phosphorus, zinc, and so on. The second pattern is called antioxidant, vitamins, and fiber. And the third one, that I think it's the easy one to establish the name, since it's really high in saturated fatty acid, monounsaturated fatty acid, and polyunsaturated fatty acid, is called fats. And then, just for the US study, the multi-study factor analysis identify one extra study-specific factor. And these study-specific factors, there are two main things. Of course, all of them are really high in calcium, and all of them are really negative in niacin. And so the nutrition tool means that this can be due to a different breakfast type that the United States population can have from the European one. But of course, they have also their own characteristic, like in MSKCC, we have some orange here with the riboflavin and so on. So they also have some differences. We also provide a validation for these dietary patterns, and our validation consists on doing a logistic regression for each of these dietary pattern, and also putting together all of these dietary pattern. We can do the logistic because this is a case control study. So we have 0 and 1, and the cases are for these oral and laryngeal cancer. So I just will describe the pattern that are significant. So the animal product and cereal is significant, and it will increase the probability of having laryngeal cancer. The antioxidant, vitamin, and fiber decrease the probability of having laryngeal cancer. And the fats has a double behavior. So it will reduce the probability and the risk of having oral cancer, but it increases the probability of having laryngeal cancer. And finally, for the Los Angeles and Boston, like we have for the same cancer site, a sort of double behavior. So like the dietary pattern of Los Angeles will reduce the probability of having oral cancer, but the dietary pattern of Boston will increase the probability of having oral cancer. So this is a way to show that like this model is not only important in the genomic application, but can be also applied to any different consortia, like for example in this nutritional epidemiological data consortia, where they wanted to check commonalities and differences. I think that this is really important also because we validate our like patterns, and we so assess their validity by putting these patterns in a logistic regression and see how they are associated with the cancer site. Questions so far?
I don't see any in the chat now.
Yeah. Okay, thanks. So the second part is regarding high dimensional data. And so what do I mean for high dimensional studies? So what we have, and this is especially like true in these genomic applications, we have some sample size with a lot of genomic variables. And so we have a setting where the number of variables P are greater than the number of the individuals, the sample size in each study. And of course, we cannot apply like the frequentist way, but generally people put some priors or penalty to add some sparsity, because otherwise we will have too much signal. And so as you can see, this is exactly the heat map that I showed you at the beginning. Right now, what you have is really high rectangular, and these other rectangular are really close to zero or are exactly zero. So in this way, we can better identify networks. And in this case, I put here common networks. So what I use here is a multiplicatory gamma process shrinkage prior. That is a prior that is coming from Bhattacharya and Danson. This prior is actually shrink all the element in the loading matrix toward zero. And actually this sparsity increase in this matrix as the column index increase. And so these are the characteristics of the prior. So we are introducing many factors. As I told you, like the degree of shrinkage increase as the column index increase. And this is quite true in the big data. And if you think about genomic, if you add more factors at the end, you will not find anything. You will not find any signal remaining, adding extra information. So this is like quite true and quite similar to the truth. And we are using for the algorithm fast and efficient Gibbs sampling. So we have also fastness. So we are detecting right now the factor loading matrix with this singular value decomposition. So as you remember, like each covariance matrix can be decomposed, each study can be decomposed in three quantities, one coming from the common, one coming from the study specific, and the other one, psi-s, coming from the error term. And so I detect these as extracting the eigenvalue and eigenvector for each of these quantity, and detecting both phi and both how many common factors we will have. And this is also an important point, because actually we can use this quantity here, as I will show you later, for networks. So also here I have a couple of applications that I want to show you. The first application is with breast cancer data. And we have in a microarray genomic context. So what I have here is, of course, again, different studies with different adjuvant therapy. And of course, again, I have different sample size. And the Bayesian multi-study factor model, right now, I am including like more than 6,000 genes. So I have more than 6,000 variables. Like right now, I'm estimating 8 common factors that actually provide the 5% or more of the total variance explained. And in total, the common factors are explaining 83% of the total variance. So I have a good explanation of the total variance of the data. So again, I will show you the heat map. Right now, I have 8 common factors. So these are just like the 8 common factors shared among all the studies. And I reperform the gene set enrichment analysis, that again is a method to assess if a pathway, so if this column here is significantly enriched in a well-known biological pathway. And I wrote here the main important one. So what I found is that the first common factor is significantly enriched in this here pathway that is really important for tumor and breast tumor progression. The second common factor is significantly enriched in the cell cycle. And as you may know, cell cycle play a very crucial role in the progression of cancer. Then I have the 5 common factors that is related to collagen. and then I have ribosomal protein, and final I have the immune response. And this is just our metrics of the common factor loadings. But we can also assess, as I told you at the beginning, the gene network. So what is, first of all, the gene network? The gene network is an undirected graph where each node represents a gene and each edge represents high expression between these two genes. And we can take actually this part, for example, so the common part of the covariance matrix, and we can assess this gene network. And we can include edge between two genes if this covariance matrix is greater or equal than 0.5. So this is our gene network is, and remember that this is just for the part of covariance matrix that is common across all the study. So I found 5 different clusters, and these clusters are exactly the same that I found before for the gene set enrichment analysis. So if you see this cluster, these clusters here has a lot of genes that are actually in the ER pathway. The second cluster has many genes that belong to the cell cycle. The third cluster for the immune response. Then I have the fourth cluster for the collagen, and the 5th cluster that is related with this, again, ribosomal protein. So I found exactly the same signal that I found before for the common factor loading matrix. So I have a second application for the multi-study factor model. And here, as I told you at the beginning, is considering tissue as a study. And this is coming from the GTEx project. So considering these, it's of course really challenging because right now what we are considering is different tissue, and of course genes like can show a diversion and diversity in expression level, revealing some important tissue heterogeneity. And this tissue heterogeneity can be really important for tissue development and some function of each tissue. And recently, like many different papers are discovering that tissue network and tissue heterogeneity can improve drug target. And this is because genes in tissue specific pattern are not expressly uniquely. And the second, because tissue network promises to be like a rich source of biomarkers for disease diagnostic and treatment as well. But there are to be consideration to be to do. First of all, that sometimes oversimplified model can miss important characteristic of this tissue specific pattern. And again, we want also to validate this result as we have done for the nutritional epidemiological paper. So we are proceeding in 2 different ways. We are applying in this GTEx project the Bayesian multi-study factor model to identify just the tissue-specific pattern. So right now in this project, it's a little bit different from the other one because we are more interested in the tissue-specific network. We are more interested in this study-specific, in the heterogeneity of the study. And we also validate this tissue-specific structure via eQTL analysis. So what is this eQTL analysis? This eQTL analysis is a powerful tool to associate and to identify an association between a SNP and gene expression. What is a SNP? A SNP is a change in the DNA sequence. So supposed to have this DNA sequence, you can see that like this G will become a T. And this is so-called a SNP, or also a mutation in the DNA sequence. So generally, like people perform linear regression in order to test the association between a SNP and a gene. but of course they are performing different linear regression in each tissue. So they are not removing the common component. And sometimes this common part, this reproducible part, can impact and can obscure this heterogeneity of the tissue. So we are proceeding with these two steps. So first of all, we are performing this Bayesian multi-study factor analysis, and we are detecting tissue-specific gene network. So right now, again, like we have the decomposition of our covariant structure, and we are actually just taking a look to this tissue-specific network, so to our lambda S. And second, we apply a regression model. So we are performing an eQTL analysis, just considering the tissue-specific part. So I'm considering 30 different brain regions in this GTEx consortium, and I will show you the result. So first of all, I will not show you like 13 different networks, but I wanted to show you just two, and I choose the important one, or the most like important one. This is the common, so I will not explore the common here, because like in the eQTL analysis, we are more interested again in this tissue heterogeneity. These two like specific network are basal ganglia, that is related to emotion and movement, and the second one that is the hippocampus, that is related to the memory, so especially long memory term and short-term memory. And this is the first area and region that is impacted by disease like the Alzheimer. And so both of these two regions are significantly enriched with this axon guidance pathway. And this pathway is really important since it's related to pancreatic cancer and Parkinson's disease. But of course, these two regions have also their own biological pathway connected. So for example, basal ganglia is enriched with the signaling by GPCR. And this is a very important pathway because it regulates many different activity in our body, like regulate hormones in the pancreas, sleep, rhythm. And the hippocampus instead is significantly enriched in this calcium pathway that is really important in Parkinson and Alzheimer's disease. And then I perform the eQTL analysis. So again, I want to see in each of these networks if some of these genes are related with a SNP, so are associated to a SNP, is associated to a mutation. And what I discover is, of course, here I'm reporting a picture. So like we have like other different eQTLs, but there are 3 genes that are related to a SNP. So the first, that is this gene called ADORA2A, that is related also to visual memory. The second, associated to the SNP, is connected to the Alzheimer's disease. And the third one, that is the MIPE, that is related to depression. So also in these like data application, we discover that first of all, it's important to estimate like both the reproducible part and the study-specific variation. And right now in this application, again, we are more interested in the study-specific component, and we want to remove this common signal that sometimes can obscure these tissue heterogeneity and these tissue patterns that are really important and connected with this eQTL analysis. So in summary, like these multi-study techniques is the only supervised analysis that provide the statistical solution to this issue of reproducibility. And again, we are able to simultaneously estimate these common and also study-specific component that both can be important in many different applications. And we have seen like the genomic, we have seen the diet, but in all of these applications, sometimes it was important to estimate just the common component and to remove the study specific that lacks cross-study reproducibility. Sometimes it's important, as I show you in the last application, to just estimate the tissue heterogeneity and remove instead the common characteristic. And sometimes, like in diet, it could be really important to estimate both. And I report here Galileo that say that his results have been obtained after the repetition or a full 100 times. And so sometimes the reproducible component and to like offer and increase also the power of the analysis that we are going to do. So the methods can be applicable to many different consortia and data, where the aim again is to assess reproducibility and heterogeneity across different studies. I want also to align that I consider study, but there could be also some occasion where you have one data and you want to assess commonalities and differences across different groups in the same data set. And I developed this MSFA package in order to make my result reproducible in many different consortia or population. And again, this is also the methods that I describe here allow to borrow power, so to borrow strength across different studies. So the reproducibility component can be really important in this occasion. And I want to highlight this reproducibility across different studies, because of course, especially in this big data world, the reproducibility is a very, very sophisticated concept, and is not as Galileo described to us, and so it can be really important to address reproducibility in many different directions for different studies. So I want to thank all of my collaborators, some from Harvard, some from Princeton, and some from Europe as well. And I want to thank you all for attending this talk.
Yeah, thank you, Roberta, for an excellent talk. We have plenty of time for questions if there are any. There is one panelist that has their, yeah, Chris, do you want to just, I don't know if you can unmute yourself or not, but feel free to.
Yeah, can you hear me?
Yes.
Okay. Roberta, that was a great talk. I think you know what question I'm going to ask you, but anyhow, so you talked about how you could apply this to groups of individuals in studies and so on. I'm wondering if you can, how you might be able to apply them to individuals themselves. Is there a way that you could relate this to people?
Yeah, actually, this is a great question, and I didn't know what question was that actually. So,
well, given that I'm always doing NF1,
yeah, but yeah, you can apply to people as well. Right now, the multi-study factor model is constructed that you should have same variable or same subject. So like you can exactly like switch the problem, and instead, for example, to have a cluster of gene, you can have a cluster of subject in each group, and of course like common characteristic across subject. But in this case, like the subject should be the same across the different groups. So it could be, for example, like, I don't know, different conditions or, sorry, same condition and maybe different variables. We are thinking also to generalize that where you can have different variable and different individuals. And that could be like also a nice direction where to go. The model right now has, it is constructed like, and also in genomic, I saw many different times, like instead of having a cluster of variables, you can have like, for example, cluster of patients, and it will work.
So I mean, I'm assuming that with your Bayesian model, you could you could sort of impute or shrink to people individually if you had enough information about them.
Yes.
Yeah. Okay. Because that would be, I would think that would be a great clinical application. If you've got somebody in front of you with specific characteristics, you might want to know what's the probability that they have a certain type of condition.
Yeah. Actually, this could be, that's a very nice, point of view. I didn't think about that. I was more thinking about, I don't know, different applications, but it's true that, also these can be applied to patients. Thanks, Chris.
Any other questions? So I just have a very simple question. In the Bayesian version of this, in the second part of the talk, is one of the ideas to sort of force some regularization on the common factors loading? Is that what you're trying to do?
Yes, exactly, something like that. Because otherwise I could not have an estimate, especially in the p greater than n. Estimate. So like I went in the Bayesian direction, but of course, Jan, as you said, one can also think about penalization and adding some penalization in both common and study specific pattern. And that could be also another like, way to go.
Oh, yeah, thank you.
Thanks.
Are there any other questions? I don't see any in the chat, but if any of the participants have any, please add them to the chat. Okay, I don't see any questions, so I think it's probably just a good idea to wrap it up. Thank everyone for attending, and thank you, Roberta, for an excellent talk. And like you can see on this slide here, the next component of this series is on October 13th between 4 and 5, and Yi Shang is going to present then. So looking forward to that. And thank you again, Roberta, and thank you, everyone.
Thanks. Bye.